Hi, everybody. It's your AP Biology teacher, Mr. Poser. Today we're getting into topic 5.4, which is in Unit 5, and this is on non-Mendelian genetics. So our last one is on Mendelian genetics, and we talked about dominant alleles, and we talked about recessive alleles, and how dominant, you only need one dominant allele to express the dominant phenotype, and you need two recessive alleles to express the recessive phenotype. And that's pretty, cut and dry. It's pretty simple. But here's the deal. Most traits that we're talking about don't really follow those rules. A lot of the genetic traits that we're going to be discussing from here on out, or that you just think about in general, are not inherited through simple Mendelian genetics like that. There are a few human traits that are like that, like, for example, like hitchhiker's thumbs or widow's peaks or that kind of stuff. But, most traits that are going to be discussed here are not going to be just determined by one gene that's associated separate, or that is assorted independently. And, it just kind of breaks the rules a little bit. So we're going to skim the surface of what's called non-Mendelian genetics. And that title screen had a fruit fly on it, and you're going to see why that is in just a second. So what I put up here is Mendelian genetics can help us make predictions about patterns of inheritance and phenotype ratios sometimes. All right. If genes are independently inherited and, the influence of one gene, the inheritance of one gene does not affect another gene, then yeah, it works all the time, right? So like with these pea plants and the flower color, it worked like a charm, right? We had two purple flowered pea plants, heterozygotes, and when we crossed them, we got a nice 75 to 25% ratio, and you'll get that every single time, okay? But that type of prediction is not always going to, it's not always going to work. We're not always going to end up with that 3-to-1 ratio. So just throwing this out there here, what if we got like 47% purple and 53% white? Then we would say, okay, maybe it isn't just dominant and recessive. Maybe there's something else going on here that's affecting the ratios at which the offspring are inheriting these particular phenotypes. All right, there's something else going on here, and that's what we do in science, right? We here's a test. This is what I think is going to happen. This is a hypothesis, and we're going to figure out whether or not it's right. And if it's wrong, we're going to figure out why it's wrong. Okay, and that's what science is about, or one of the things that science is about, right? And I'm going to show you an example when we were wrong coming up here, okay, or when Mendelian genetics is wrong, I should say. Okay, so lots of traits. My point is that they don't follow Mendelian patterns. Okay, genetics can get a lot more complicated. All right, and we're going to skim the surface here today. For example, like human height or something like that, right? That's determined by like 200 different genes, not just one gene. Okay, so there you go. All right, if height was determined by one gene, it would probably just all be two different heights. You'd either be tall or short, something like that. I don't know. But my point is that it gets complicated, and so we're going to talk about several different ways that the Mendelian laws are kind of broken. All right, independent assortment is not really working. Dominant always doesn't mean dominant, and recessive doesn't always mean recessive. All right, so here we go. We're going to look at some other inheritance patterns too. So the first example of, Mendelian genetics being wrong, the last video, kind of just saying, oh, here's dominant, here's recessive, this is what it means. This is our first one. It's called codominance, and it's basically when two alleles are both dominant at the same time, okay, and they're both expressed. So like, for example, if we're talking about the pea plants again, right, if you have a purple flower and a white flower and you cross them, and the resulting offspring have both purple and white flowers, like both at the same time, then that might be codominance. So I put codominance occurs when both alleles are expressed and heterozygotes have a different phenotype than both types of homozygotes, okay? So like if both are being expressed, all right? Another good example is in human blood type, right? There's 4 different human blood types. There's only 3 alleles for blood type, but there are 4 different types of blood type, all right? So let's just say if your mom is type A and your dad is type B, that means they have these little antigens on the surface of their red blood cells. Hey, we don't have to get into the specifics about it, but let's just say your mom's type A and your dad's type B. You have a possibility of being type A or type B, but you also have a possibility of being both at the same time, and that would be type AB. You can see both of these proteins here present on that red blood cell. Okay, so AB is a different phenotype than A or B, and this would be an example of codominance, A and B both being dominant at the same time. Okay, kind of the complete opposite of codominance is incomplete dominance, and that occurs when neither allele dominates the other. So heterozygotes are quote-unquote blends of the dominant and recessive phenotype, all right? So this is kind of like when neither is dominant, So we're using capital and lowercase letters up here to represent dominant and recessive, but neither of them are dominant because we can see, all right, if you inherit two of these capital R's, you have red flowers or red petals here. If you inherit two of the lowercase alleles over here, you have white. Hey, but check it out. One of each doesn't result in just red. It results in pink, which is a blend of the two. Okay. So that's what incomplete dominance is. So anytime you're seeing a blend of two phenotypes from the parents in the offspring, that's how you know it's going to be incomplete dominance. All right. Another example of when Mendelian genetics is wrong, it has to do with sex-linked chromosomes. All right. Some genes are located on the sex chromosomes, and inheritance depends on the inheritance of XX or XY chromosomes. Now, sex chromosomes are different in other animals and plants than they are in human beings, but for simplicity's sake, let's just talk about people, right? Someone who is biologically female is going to inherit one X chromosome from their biological mom, one X chromosome from their biological dad. Someone who is biologically male inherits one X chromosome from mom and one Y chromosome from dad. Okay, so XX or XY determines one's biological sex, okay, as you can see from this diagram over here, right? Now, check it out. If there's a recessive allele located on the X chromosome, then some interesting stuff happens, right? So, like, check it out. If these two have kids, right, and check it out, they have the possibility of producing a son without this X recessive allele. They have a possibility of producing an unaffected daughter, the possibility of producing a son that's affected by a disease, all right, and then a daughter who's a carrier, okay? Now, what does that exactly mean? I'm going to put this in a Punnett square again because Punnett squares are easy to kind of understand. All right, let's talk about color blindness. All right, color blindness is an X-linked recessive gene. All right, so being unable to see or discern particular colors from one another. There's lots of different kinds of color blindness, but I think this would be like red-green color blindness. It's determined by a sex-linked recessive allele. All right, so I put this little C up here next to the X chromosome to represent that this is an X chromosome that contains a recessive allele that causes color blindness. Okay, so here's a mom who is what we refer to as a carrier. She has one copy of the gene, but she is not colorblind, and I'm going to show you why here in a second. It's recessive, right? So she would need two copies of it to be colorblind, right? And here's dad. Dad doesn't have the colorblind gene, all right? So as I just said before, that is a sex-linked recessive, I should say allele up here, not trait, sex-linked recessive allele that's attached to one of mom's X chromosomes, right? So if we cross these two, they have kids, all right? They have one daughter who's what we refer to as a carrier. They have another daughter who is, does not carry the gene. They have one son who has one copy of the gene, and then they have another son who does not have a copy of the gene. Now, one of these kids is colorblind. Can you guess which one it's going to be? Well, it's going to be this guy right down on the bottom left. Okay, now, we just talked about in our last lesson how recessive alleles, you need two copies of it to express the recessive trait, right? Not necessarily, right? Because check it out. If this is located on X chromosome, right? Guys, men, biological men, we don't inherit two X chromosomes, right? We only get one chromosome. So if you're the son that inherits that X chromosome from mom that has the sex-linked recessive gene, well, guess what? You're colorblind, all right? So it's way more common. If you know somebody who's colorblind, I'm going to go ahead and venture and guess that they are biologically male, simply because it is going to be more common in biological men only need one copy of that recessive allele as opposed to biological women, the daughters, who are going to need two. Now, if this daughter had XCXC, she had two copies of that recessive allele, then she would be colorblind. But for right now, she is not colorblind and she is just a carrier because this is still a recessive allele. All right. So something that you're going to see coming up here are pedigrees. And you're going to have to determine, okay, what is the mode of inheritance? Is it going to be dominant? Is it going to be recessive? Or is it going to be sex-linked recessive? Or sometimes you could even see sex-linked dominant. So this is what a pedigree would look like for something that would be sex-linked recessive. Okay, so check it out. This is let's just say color blindness, all right? This mom is a carrier right up here. Here's the key, right? This mom is a carrier. She has a daughter who's a carrier. a son who's unaffected, another daughter who's a carrier, and another daughter who's unaffected, but she has one son who is one son who is affected, one son who's not affected, two carrier daughters, and one unaffected daughter. Maybe I already said that. I thought I counted her, but anyway. Hey, check it out. We can tell that this is sex-linked recessive, okay, because, well, it's more likely that the males are becoming colorblind, let's just say here, and the females are not colorblind. They're going to be carriers, and you can follow this mode of inheritance down here, right? Here's the colorblind son. He's passing down an X chromosome to one of his daughters, and then that daughter is passing that X chromosome down to one of her sons. You can pretty clearly see that pattern here of sex-linked recessive inheritance. Okay, so I'm going to show you one more pedigree here where you might have to look out for what mode of inheritance it is, but we'll get to that in just a second. All right, another example of when Mendelian genetics is wrong is a pleiotropy, and that is when one gene affects multiple phenotypes. And traits do not segregate independently. So lots of genetic disorders, they're usually resulting in one mutation in one gene, but that one mutation affects more than one trait, right? So that protein that's being produced from that faulty gene has multiple maybe functions, and therefore it results in several different alterations rather than just one gene for one trait. That's called pleiotropy, and this is what this is trying to show you here. One genotype results in multiple phenotypes. That's a thing. All right, that's called pleiotropy. The last one before we get into the big one here is what's called mitochondrial inheritance. So what we've been talking about so far is, DNA that's passed from parent to offspring, right? That's very important. But I failed to mention that is DNA from the nucleus of the cells, all right? And remember, mitochondria are their own little things, right? Remember endosymbiosis? They have their own DNA. They have their own proteins, right? So here's the deal with that, okay? Mitochondrial DNA can carry mutations. It can carry different alleles as well, okay? So mitochondrial inheritance involves genes located in chromosomes, located in chromosomes in mitochondria. Oh, I should add that in there. That's a typo, okay? Chromosomes in mitochondria, okay? Just add that. Wow, handwriting super sloppy, okay? But it's only passed down maternally, okay? So here's the deal. Your mitochondria that your cells have, well, you got them from mom because egg cells contain mitochondria and typically sperm cells do not, all right? The same deal is with chloroplasts. Chloroplasts only come from the egg of the plant and not from the pollen, from the sperm, essentially. So yeah, check it out. This pattern of inheritance is different because check it out. If mom has a mitochondrial disorder or some kind of allele that's only in her mitochondrial DNA, all of her kids are going to get it no matter what, right? Because their mitochondria only come from mom, all right? The reverse is true as well, right? So if dad has a mitochondrial disorder, none of his kids are going to get it because he's not passing down his mitochondrial DNA to his kids. It's only passed down maternally. All right, so here's one more pedigree that demonstrates mitochondrial inheritance. All right, and something that you can tell here is that, here's looks like great grandma up there. She has a mitochondrial disease. All of her children have that mitochondrial disease. All right, and therefore, check it out. This mom with the mitochondrial disease, all her children have it. All of her children have it. Okay, but check it out. Here's a dad with this mitochondrial disease. None of his children have it. Okay, same over here. So this is very clear here that this is mitochondrial inheritance because it's only being passed down from mothers. It's only being passed down maternally, and mitochondria are only passed down from mom. All right, now here's the big exception to Mendel's rules here, okay? As I said before, Mendelian genetics assumes genes are assorted independently and randomly, meaning the inheritance of one gene doesn't affect the other, but this isn't always necessarily true. Now, there's another very famous set of experiments besides, not quite as famous, but still very well known besides the pea plants. It involved this guy over here. His name was Thomas Hunt Morgan, and why I had the fruit fly on that initial slide is because, well, he studied fruit flies instead of crossing pea plants to determine their modes of inheritance. He's studied fruit flies. Fruit flies are pretty easy to cross. You can probably do an experiment crossing fruit flies in your AP biology class if your teacher has the means to, grow fruit flies and have you guys sort through them and that kind of stuff. Most AP biology classes just do the virtual fruit fly lab, but I digress. Here's the deal, okay? I'm going to try and keep this brief here. He's crossing fruit flies, right? And he's crossing fruit flies with two different traits, right? So he's kind of doing dihybrid crosses a little bit. All right, and one of the more famous crosses experiments that he had was when he took this fly over here, which was what's called wild type, okay, and crossed it with another fly that had two what are called mutant traits, all right, which are not typical of what you would find in the wild, right? So wild type just essentially means normal, what you would see on a regular fruit fly, and then these mutants are, well, they have mutations to certain traits that separates them out from typical wild type flies, right? So you have one wild type fly that's heterozygous, right? So I'm going to just tell you right now, I'm going to spoil it a little bit, that the wild type traits are dominant here, okay? And wild type traits are indicated by this plus sign over here. This one has two mutant traits. So it has a black body and something called vestigial wings. Okay. Now I tried my best to try and crop out the wings and make them smaller, because that's what vestigial wings are, but I don't know. I don't have the photo editing skills for it. So maybe someday, I think I've done better with my animations and stuff with these videos, and make that a little, cleaner, but yeah, I don't know. I tried to get rid of these wings, and it just didn't work out that well. So anyway, here's a heterozygous fly. It's wild type, which means it has the normal traits. And here's one with two mutant traits, okay? So these plus signs, you can kind of think of them as like capital letters, right? They're dominant. So like VG plus, that's a dominant allele. B plus is a dominant allele, while VG is a recessive allele and B is a recessive allele. All right, VG is the gene that's going to determine vestigial wings, the absence or presence of these shriveled up wings, like this fly has over here. And then B is going to be determining its body color, essentially. All right, so this one is brown, which is like the regular color of fruit flies because it has one of these wild type alleles, and this one is black because it has two of these recessive alleles. Okay, now if we cross these two flies, all right, let's set up a dihybrid cross here. All right, you would have to do your FOIL method once again. All right, and we got our dihybrid cross set up already for us, but if we did our FOIL method, right, we'd have one possibility where both wild type alleles, one or the other is getting passed down, and then neither wild type alleles are getting passed down, right? But the combinations of alleles up here are going to be the same, right? Because they're only going to be passing down the recessive alleles. All right, so at this juncture here, pause the video, work this out. What is the phenotypic ratio that we can expect? How many flies out of the total are going to have black bodies? How many of them are going to have vestigial wings? How many of them are going to have both, neither, or one or the other? Okay, let's try to work that out. All right, if you're ready to go, I'm going to go ahead and click next here. All right, and if you worked it out, this is what it should look like, okay? You should get one row of wild type flies, okay, regular body color, regular wings, one row of black flies with regular wings. You should have one row of brown flies or wild type color flies with vestigial wings. And then you should have one row with both black body and vestigial wings, okay? So you should get in other words, 1 to 1 to 1 to 1 phenotypic ratio. Each of those is going to be 25%. That's what we should expect, okay? So when Thomas Hunt Morgan was doing these crosses, he's like, okay, we should get 25% of this phenotype, this phenotype, this phenotype, and this phenotype. All right, based on the laws of probability and the predictions. All right, so here's the deal. Well, I want to give you one kind of vocabulary term here. 50% of the flies are going to match the parental phenotypes. Like this row right here is matching the parental phenotypes, and this row right here is matching, right? Because we have a wild type for both, and then mutant for both down here. Okay, but these other two rows in the middle are what are called recombinant, and they have a new combination of traits that are different from their parents. Hence, that's a result of crossing over, which we talked about in 5.2. Now, here's the thing, all right? That's what we expected, but take a closer look. Okay, when Thomas Hunt Morgan crossed these flies and collected all the offspring and analyzed their phenotypes, it turns out only 17%, 8% of one category of flies, and 9% of another category of flies, the recombinants, were present, okay? And a much higher number than 25% were found for these what are called parental phenotypes. So, as I kind of mentioned at the beginning of the video, all right, if you're not getting your expected percentages, something is going on here, all right? It's not following Mendel's rules, it's not following the regular rules of probability. So, what's going on here? And this is the big exception. This is the big one. There was less than 50% recombination between vestigial wings and black body because the genes for body color and wing shape are located on the same chromosome, or in other words, they are linked. Okay, they are linked on the same chromosome. Okay, in other words, they are more likely to be inherited together than they are to be inherited separately from one another, split up from one another, and so to speak. Okay, so we only got 17% recombination here. All right, different from the parental phenotypes, and that's showing us here that if it's less than 50%, then these two genes are located on the same chromosome as one another. All right, and therefore that flies in the face of the idea that, hey, genes are independently assortative from one another. All right, there's the inheritance of one gene doesn't affect the inheritance of another gene. That's not true if they're located together, and they're more likely to be inherited together than apart. Okay, and this ties back to us crossing over. All right, we talked about crossing over in 5.2. As I put here, genes close together on the same chromosome are less likely to be crossed over and more likely to be inherited together, right? So check out B and C over here. All right, this top scenario over here, okay, is more likely to happen than this bottom scenario over here because B and C are nice and close to each other, okay? This type of crossover here where B and C are basically like swapping places and getting switched over like that, recombined, is less likely to occur than if both of them got switched over together. All right, so the comparison lies in right here. Okay, recombination results in different phenotypes than the parents, right? And that results from crossing over. All right, and if they're closer together, they're more likely to be crossed over together rather than crossed over separately. That's what I'm trying to say here. Okay, so since Thomas Hunt Morgan got 17% recombination, 8% of one phenotype, 9% the other phenotype, that means that those two genes are 17 what are called map units apart from each other, or as I like to call them, centimorgans. That's the other, the unit that's used for the distance between genes on a chromosome. You know, if you made a discovery like this, I think, or if I did, I would probably want to, name that unit after myself. How about centiposers? That would be awesome. 17 map units are centimorgans apart from each other on the same chromosome, okay? If there was 28% recombination, there'd be 28 units apart. If there was 39%, all right, there'd be 39 units apart. But if it's more than 50% recombination, they are independently assorted, all right? The inheritance of one gene does not affect the inheritance of another gene, or they're located separately on separate chromosomes. Okay, so there you have it. All right, if you're finding anything less than 50% recombination on a cross between a heterozygote and a double mutant, then those two genes are linked. They're located on the same chromosome. All right, and Mendel's laws are not really applying. Independent assortment does not really apply when it comes to these genes. Okay. So yeah, there you go. All right. Just to wrap this up here. All right. And Thomas Hunt Morgan and his team kept on doing all sorts of different crosses here, and were able to essentially put together a map of a chromosome of a fruit fly. All right, indicate how far apart and how close together particular genes are. All right, so if you do a bunch of these crosses over and over again and you determine recombination percentage, you can make a map of where these chromosomes are. All right, so just a super easy example. All right, something you might come across in class. Let's just say I have a gene E, all right, maybe for like eye color or something like that. Had 25% recombination with VG and 8% recombination with B. Where would it be located on this chromosome? Well, if it has 25% recombination, it's 25 centimorgans away from VG, and if it has 8% recombination, With B, that means it's 8 centimorgans or, map units away from B. So it would probably be located right there, okay? 25 away from VG and 8 away from B. So it would be located right there. All right, and that's how you can make gene maps. All right, so let's recap here just in case you missed anything or I missed anything. I'm just going to briefly run through these. Mendelian rules are not always followed. There's always lots of exceptions. Independent assortment of genes is not always how it works, okay? There's incomplete dominance. Neither allele is dominant. Codominance is when both alleles are dominant. All right, that was with like the blood types. Incomplete dominance was the flower colors, the pink flowers. Sex linked. All right, we looked at color blindness. We looked at a pedigree, and we determined that the inheritance of recessive alleles on X chromosomes is going to show a different pattern, right? Biological men are more likely to inherit X-linked recessive traits, phenotypes, because of sex-linked genes. Mitochondrial inheritance, we looked at a pedigree of that. Okay, that's when genes are located in mitochondrial DNA and are only passed down from mothers to their offspring. It's only passed down maternally. We looked at a pedigree of that. All right, pleiotropy is that last small example. All right, one gene affects multiple phenotypes. That kind of bends the rules of Mendel as well. All right, but the big one that we just spoke about was linkage, where genes are not inherited independently. They're located on the same chromosome, and they're more likely to be inherited together than they are separately from one another because of crossing over. Okay, that linkage is indicated by anything less than 50% recombination from a heterozygote and double mutant cross, like what we did with that dihybrid cross that we worked out. And recombination frequencies can be used to make gene maps of a chromosome, right? So like 27% recombination means 27% or 27 map units are sent to morgans apart from each other. You can make a map that way. It's kind of cool. All right, that's it for this video. I know it's another long one, but there's a lot to talk about. And I think in my last series, I separated out these into 2 videos. So I think I spent less time on this. So yay. Let me know if you have any questions. We'll see you next time.